Good afternoon, students, and how have you been doing? I hope you are well. It's 3 p.m. now. We will start soon our problem solving session. Let's wait for 5 more minutes so that some more students can join us. And as you guys know, we'll be solving practice problem lecture week 4. So these problems will be corresponding to the lecture 4 of the series. And I hope you guys are going through your lectures uploaded by NPTEL. Go through them, please follow them, and attend the TA sessions so that you'll have a better understanding of solving the questions. And the general instructions again to all the students. So keep your mic and video chat turned off during the whole session. You can communicate with me and answer the questions by typing in the chat box. That will be a mode of interaction between you and me, and we'll solve a similar set of 10 to 11 questions which are corresponding to lecture week 4, and it will be kind of similar to what you'll be facing in the assignment. So hope that today's session will be helpful for you guys. We'll start at 3:05, and meanwhile I'll keep admitting students who are trying to join this call. So it's already 3:05, and I've admitted the students. So if more students keep joining this call, I'll keep admitting them. So now I'll share my screen. So we'll have a set of 10 to 11 questions, multiple choice questions, which may have one or multiple choice answers correct. as you must have solved the assignments of the previous weeks, and we'll go through them one by one. I'll post the questions for you and read it out for you, the questions and the options. And I expect the students to understand the question, and then just let me know in the chat box that what do you think is the correct answer for this question, for the question that will be posed on the presentation. It's okay if you are able to answer it correctly or not. It's fine, do not be shy. And we'll be discussing the answers of this question as well as a brief concept about what the answers are. So that's how we'll solve 10 to 11 questions through this session. So let me just share my presentation. I hope you are able to see the slides clearly and my voice is clear to you also. And can you see the slides changing? Just let me know in the chat box with a quick yes or no, then we'll begin shortly. If you're able to see my slides and my voice is clear to you, great. Thanks, Sakshi. So good afternoon to you all again, and welcome to problem solving session for week 4 lectures, and we are in the course together for Introduction to Developmental Biology. The course instructor is Professor Subramaniam from IIT Madras, and my name is Ashutosh. I'll be the teaching assistant for you guys for this session. I'm a PhD scholar at IIT Kanpur. So another of my friend who is also a TA for this course is Parth De, and Parth will be having his classes on Monday evening. He's from IIT Hyderabad, so he'll be solving some questions. So with that much introduction, let's move on to the first question. The first question corresponding to lecture week 4. This is a true-false question. Which of the following statements are true? Mind you that multiple statements can be true as per the instructor. So you have to go through each statement and tick the answers which you think are true. Statement number 1, embryonic cell division in C. elegans varies from worm to worm. Statement 2, Lin3 is required for induced fate in vulval formation. Statement 3, variable number of tandem repeats can act as molecular markers for mapping. And statement 4, in sequential substrate enzyme pathways, mutation in a gene is epistatic over mutation in a gene that functions upstream. And the last statement, mosaic analysis helps to identify spatial and temporal constraints on gene activity. So these are the 5 statements here, and now it's up to you guys. What do you think? Which of these statements are true? So if I want to give you a hint, all these concepts are taught across the whole 3 lectures in the week 4. So one or two information from C. elegans, the other from uh, polymorphisms, variable number of tandem repeats, epistatic pathway. So you guys, if you guys have been through these lectures, you will be able to answer. It's okay. I want all the students to try and participate in this problem session. So let's take a minute and collect some answers before moving on to the correct answer. Which of these following statements do you think is true? Sakshi says 3 and 4. Okay, We'll check at the end what is the correct answer, and we'll understand some of the concepts of this correct answer. We have another answer. Ajay is saying 1, 2, 3, and 5. Okay. Any more answers? Ashwini, Pushpanjay, and Simhasanam. What do you guys think? Do you or will you be able to answer? You can just even guess based on the statements and your logic what you think would be the correct answer. 1, 3, and 4. Cool. Okay. Without any further delay, let me reveal what are the correct answers. The first correct answer is statement 2. Lin 3 is required for induced fate in vulval formation. This is a true statement. The second correct answer is variable number of tandem repeats can act as molecular markers of mapping. So statement 3 is true, which I think Ajay, Sakshi, and Simhasanam have all answered correctly. And the third correct answer is mosaic analysis helps to identify spatial and temporal constraints on gene activity. So these three statements are true and are the correct answer to question number 1. Now let us throw some light on these answers. So the first statement, no, the C elegans is referred to here as worm. So C elegans is a worm. So embryonic cell divisions in C elegans. Yeah, yeah, but yeah, you're correct. You're correct, Ajay. But let me just clear your concepts. Here we are what the question has asked is embryonic cell division. So what you are talking about is chromosomes, right? Segregation of chromosomes. But here what we are talking about is embryonic cell division. So how from a zygote the worm is forming? How from a zygote multicellular worm is forming? So you will be clear here. I think you got confused in the statements. Yeah, yeah, yeah. Actually, from the zygote we have fixed number of cells. So in the C elegans, we have mapped everything, right? That's what Professor Subramaniam has also taught. That yes, you are correct. And each cell have already we have known what it is going to form. So the type of cell division is same in C elegans. The chromosome segregation might be different. Yeah. Okay. Okay. So XX and XO will occur in the germ line of the organism, right? So here, if you see here, so if you see this, look at this map here. From zygote, we have AB, P1, all different kinds of cells, and the embryonic division is patterned. So what you are trying to say is XX and XO will occur in the adult life of the worm, right? So it might not be embryonic. So that will be my point for distinguishing. So after the germ line is formed in an adult organism, now the organism can uh, undergo division to form XX and XO. So the embryonic cell division refers to development from zygote to an adult organism, or the different types of cell types that we are seeing here. Like neuronal muscle come from AB cell type, while P1 we have asymmetric divisions. But these divisions happen in every organism. So may it be XX or XO, from the zygote it will develop similarly as a germ line. Then whatever the chromosome composition is in the germ line, they can form gametes. XXO will form one X gamete, and XX will form two X gametes. That's how. I hope it's clear to you. So the embryonic development of every worm will be same. That is the point one. The question one. Embryonic cell division in C. elegans varies from worm to worm is not true. I hope it's uh, I hope it's clear to you. Okay, okay, cool. Yeah, as I was saying, so the development from the zygote to adult organism, the worm has fixed number of divisions. Even the timeline is mapped. Now we know everything about the C. elegans worm, and there are fixed number of cells, and we have different kinds of cell giving rise to different lineages of tissues found in the C. elegans worm. Embryonic cell division in C. elegans follow a strict pattern, and fate of each cell is predetermined. This means that in every individual worm, the same cells are produced at the same time and in the same order. So every worm, the same cells will form, are produced at the same time and in the same order. So AB will form ABA and ABP. The zygote will first asymmetrically divide into AB and P1. ABA, ABP will form the hypodermal, neuronal, and muscle and excretory systems, while P1 further undergoes asymmetric division to give EMS and P2. EMS will give rise to muscle and E for intestine. P2 will give rise to C and P3. So similarly, we have lineages derived from each cell type. And here is the timeline in which these cells divide. So this pattern is fixed in a C. elegans. So it will stay same in the word. Now the second statement was about Lin3. So here we see the one of the most studied phenomenon of vulva development in C. elegans. So in vulva development, we have a cell called anchor cell. The anchor cell is a specialized cell located in the posterior region of the C. elegans, in the gonad of the C. elegans. And during vulval development, the anchor cells play crucial role in inducing neighboring cells to adopt specific phase and ultimately form the vulva. So what the anchor cell will do, it will induce its neighboring cells, the cells that are adjacent to the anchor cell, to specific fates, and it will form, collectively they will form the vulva. Ultimately they will form the vulva. And one of the major important factors is Lin3. The Lin3 gene encodes an epidermal growth factor like protein in C. elegans. So if you remember, there are many signaling pathways which are very important for vulval development, and one of the most important signaling pathways is EGF, or epidermal growth factor like uh, pathway, which then with the help of RAS, RAF, MEC, ERK signaling produces the vulva. So it's one of the important factors, and Lin3 is a gene which encodes EGF like protein in C. elegans. And the Lin3 gene product is a signaling molecule secreted by the anchor cell. So the anchor cell will secrete Lin3, which will then influence its neighboring cells to become the vulval cell. And when the anchor cell comes into contact with certain cells in developing vulva, it releases the Lin3 protein, which then binds to and activates EGF receptor on neighboring cells. So these neighboring cells have EGF receptor on them, and Lin3 is the ligand. Lin3 is EGF family protein. The EGF signaling will turn on in the neighboring cells, which will induce vulval development. So statement 2 is correct. Lin3 is involved in vulval induction. Then statement 3 is about VNTRs. So what are VNTR? VNTRs are expanding to variable number of tandem repeats. It's given in the statement 3. And VNTRs are polymorphic, meaning they have many forms and the number of tandem repeats of a sequence is highly variable within a population. So VNTRs are some repeats which are unique to an individual, which can act as a marker for that individual, right? And while repeated sequences themselves are usually the same from person to person, the number of times they are repeated tends to vary. So VNTR are repeats of the same sequence. Like here we see, here an individual has so many VNTRs. The red is one single unit of VNTR. So any particular individual has a fixed number of VNTRs. And VNTRs can occur in the chromosome near to genes, and they act as a genetic marker for that individual. In order to create a DNA fingerprint that is unique to each person, scientists had to identify areas in the genome that are highly variable. VNTRs are highly polymorphic. VNTRs tend to differ among different people. That is what the advantage of using this as a genetic marker. The number of times the sequence is repeated can differ within and between individuals, therefore making VNTRs a polymorphic entity. So statement, I think, yeah, statement 3 is also correct. They can act as molecular markers for mapping. But for statement 4 and statement 5, in statement 4, the statement says in sequential substrate enzyme pathways, mutation in a gene is epistatic over a mutation in a gene that functions upstream. So this is false because it is the opposite way around. The mutation in the upstream gene can be epistatic over the downstream gene. So it can mask the effect of the downstream gene. So statement 4 is incorrect. And mosaic analysis help to identify spatial temporal constraints on gene activity. So in mosaic analysis, a cell uh, can can have both maternal and paternal copy. That's why it's an important. It can be used as an important assay to identify the constraints on gene activity. So let's say X chromosome inactivation was top one of the ways in which mosaics can be generated. So random, so the X chromosome inactivation will randomly suppress one of the X chromosomes in a double X phenotype. So when we have XX, the dosage compensation happens and one X chromosome is randomly silenced. And in an individual, all the somatic cells, they have random X silence. You may not find an individual with all the same X chromosome inactivated. So many somatic cells can have different X chromosomes inactivated, maybe the first one or the second one. So that brings us to these three correct answers. LIN3 for vulval formation, VNTR as molecular mapping, molecular markers for mapping, and mosaic analysis for spatial and temporal constraint on gene activity. Yeah, I hope it is clear to the students. Just let me know with a yes or no, and we'll move on to the next question. Is it clear to the students? Great, great. Okay, let's move on to the next question. So for questions 2, 3 and 4, we'll refer to this table. So for this table, we have different mutations here. So these V, W, X, Y, Z, these are all mutations with a vulval S phenotype, and crosses were performed to identify complementation groups, and the following data were obtained. Yes, Ajay, I read your comment. Great. So if you look at this table, so this is a complementation cross table, and we have different mutations over here, V, W, X, Y, Z. and these all these mutations give the vulvaless phenotype. So in this phenotype, the worm will not have the vulva. Vulva formation is impaired. Now there will be some questions based on this table. Sorry. Yeah. So which of the following statements are false? So this is the second question. Which of the following statements are false? There are two complementation groups, VW and XYZ. And the second option is there are two complementation group, VX and YZW. And the third and fourth statement, genes within the same complementation group complement each other. Genes from different complementation groups can complement each other. Now you have to identify which of these statements are false. So you just have to remember the complementation groups here in the options: VW, XYZ, VX, YZW. So I'll show the table again. VWXYZ was the first option and VXYZW. So this is the table. Look at this carefully and you tell me which of these do you think are the false statements. Two complementation groups have been identified, and what are the characteristic of genes within the same complementation group. Which genes can complement each other. So I'll make your thinking easier. So there will be two correct answers. So one from the first two statements and the other from the second two statements, the third and fourth statement, right? Which of these statements are false, which are not true? Now let us wait for a minute and collect some answers. I'll keep the table in front of you so that it will be easier for you to refer. Okay, Ajay has answered option 2 and 4. Simasanam is saying 2 and 3. Sakshi says 2 and 4. And Pushpanjay is answering 1 and 4. So you have to carefully read the question. Which of these are false statements, which are not true? Okay, let us check the correct answers. So first correct answer is statement 1. There are two complementation groups, VW and XYZ. Now let us see. So this statement is false. The first answer is VW and XYZ. VW and XYZ. These are not complementation groups. And the second answer is option C. Genes within the same complementation group complement each other. Genes within the same complementation group complement each other. This statement is also false. So 1 and 3 are the correct answer. Now let us understand about complementation. So this is a very simple but tricky concept. Even students get confused, and it's pretty easy to remember. You just have to draw it out. So here is an example showing a complementation group. M1 and M2 are two separate recessive mutations that both result in the same uncoordinated behavior. So let us consider two recessive mutations with uncoordinated behavior. M1 with M1 recessive will give uncoordinated. M2 with M2 will give uncoordinated, two copies of M2. Whereas M1 plus will give wild type since M1 is recessive. and M2 plus will also give wild types if M2 is also recessive. Now, if both of these mutations are present in a trans configuration and an uncoordinated behavior is observed. So let's say we have one copy of M1 and one copy of M2 in trans, and now again the uncoordinated is observed, then these mutations do not complement each other and are alleles of the same gene, because if you see M1, the absence of M1 cannot be compensated by M2, cannot be complemented by M2. Vice versa, whatever mutation is causing this M2 gene to go down, M1 cannot complement it. So what does that make M1 and M2? These do not complement each other. And such two genes or two mutations are grouped into what is called a complement. So in a cross, when we see that something is not able to restore the phenotype, then that is a part of the complement group. So it's very contrasting. Two genes which cannot complement each other will form a complement group. But what happens if the trans configuration results in a wild type behavior? Let's say we have something like this, M1 with wild type, M2 is on a different gene. So M1 and M2 mutations on the same gene, then we'll still have the uncoordinated behavior. If M1 and M2 are mutations on different genes, then both will be compensated, both will be complemented by the good copy present in the other chromosome, right? Then these mutations do complement each other and are alleles of different genes. So we will group such genes or such mutations in different complementation groups. So it's again tricky. Two mutations or any two genes which are not complement in the same complement group may or may not complement each other. So they can produce a wild type phenotype when they are sat together. So this is the key concept behind complementation analysis and grouping. So two mutations on the same gene will be placed in a complement group and they will not be able to complement each other because they are two mutations on the same gene or in different alleles of the same gene. So if you see the first statement here, there are two complementation groups VW and XYZ. This statement is false because the complementation groups are Vx and WYZ. So these are the two complementation groups. And how did we find it out? So all these mutations are vulvaless phenotype. We know that. Now we will cross these mutations against every other mutation one by one and see which which genotype is restoring the wild type situation, right? And which mutation is not able to restore the wild type situation. Here we see when we cross VV with XX, we see that we generate a vulvaless phenotype. So VV and XX therefore are in the same complementation group and they are mutations either in the same gene or in the same allele. That's why the vulvaless phenotype is still not restored. But it's not the case with WW, YY and ZZ because VV and WW are different genes or present on different alleles. That's why they are able to compensate or complement each other and produce a wild type phenotype. Similarly, we have WW producing vulvaless phenotype with YY and ZZ. So WYZ form a complementation group while V and X form a complementation group. So this is the true statement. Vx and WYZ are the complementation group. So that's why statement 1 is false where it was written VW and XYZ are complementation group. Complementation test is a genetic experiment used to determine whether two different individuals with the same phenotype have mutations in the same or different genes. This is the basis of complementation test. So we have two genes and they are not able to restore a phenotype. When they are not able to restore a phenotype, they are grouped together in one complementation group and they are considered to be mutations in the same gene or in the same allele. Now for the second statement. So to summarize, genes within same complement group cannot complement each other. This was the second correct answer, which was false, which said genes within same complement group can complement each other. In genetics, a complementation group refers to a set of mutations that do not complement each other. So this is a very important but simple concept to remember. And in other words, if two individuals with mutations in the same complementation group are crossed, the resulting offspring will still exhibit the mutant phenotype. So this is what we saw when we crossed VV with XX. They still produce the mutant vulvaless phenotype. That's why they are together in a complement group. This indicates that mutations are likely affecting the same gene or the same functional domain within a gene. So it might be the same gene, mutations in the same gene or same functional domain. It might be acting in the same pathway, which leads to the creation of vulva altogether. So the statement 1 and 3 will be false. Statement 1 was the complementation groups was V, W and XYZ. That is false. And the second statement, which was false, was genes within the same complement group cannot complement each other. This is a true statement, while the statement in the question was genes with same complement group can complement each other. I hope we are a bit clearer on the concepts of complementation by solving this question. So we have part uh, question number 3 and 4 based on the same table of uh, mutant mutations and complementation test. I hope question 2 is clear to everybody. Let me know in the chat box with a yes or no. It's good to see that you guys are participating and answering. It's okay if your answer is wrong. You do not need to feel bad about it. Yes. Thanks, Sakshi and Ajay. Great. So let's move on to question 3. Now, what can we say about the relationship between mutations Y and Z? Remember Y and Z? Let me show it again. What can we say about the mutations of Y and Z? Right? You remember this? Because I cannot show it on the same slide. It will be congested. They are on the same gene. They are on different genes. They complement each other. They cannot complement each other. Now, what is the relationship between the mutations Y and Z? 4 statements. Let me know that what you think is the correct answer. Y and Z. It's very intuitive after the explanation we had just now that Y and Z belong to the same complementation group, because we saw beforehand, if you see, follow the table again, Y and Z, when you cross YY with ZZ, you still get vulvaless phenotype. So now, but the statement is, what is the relationship between Y and Z, these mutations? Yeah, Y and Z. Okay, Ajay has answered 2 and 4. I think it is for the third question. And Simasanam has answered 1 and 3. You might have to look at the table again. Y and Z. And I have already given you a part of the answer, that they are in the same complementation group. W, Y, Z are in the same complementation group. Now you use your concepts about the complementation groups to find out whether they are on the same gene, they are on different genes, they complement each other or they cannot complement each other. So I'm kind of making it obvious. Guys, are you able to follow it? Then you let me know about the answer in the chat box. Simasanam and Ajay have answered already. Sakshi, Puspanjay, Ashwini, come on guys, you guys should be able to answer this. Okay, okay. Let's not waste more time, and let me reveal the correct answer. The correct answer is they are on the same gene. Y and Z are mutations in the same gene because they belong to the single complementation group, right? That's what we learned. Because Y and Z are still producing the vulvaless phenotype. Y and Z mutations are not able to complement each other. That means they might be the mutation on the same gene which is getting dysfunctional, and they cannot complement each other. So the answer would be option 1 and option 4. This is the correct answer. Yeah, Ajay, you want to speak? Simhasanam is speaking. Simhasanam, is it? Yeah, yeah. You see, can you see the table here? Y and Z. Yes, definitely. YZ belong to the same complementation group. That means they are not able to complement each other. You see here, when YY is crossed to ZZ, we still get the vulvaless phenotype, right? Right. That's why it is a high chance that Y and Z are mutations in the same gene. Are you able to understand it? Let's say we have a gene product. Let's say whatever this vulvaless phenotype, a gene is producing a protein which is responsible for formation of vulva. Now the gene has a mutation Y where the protein is not produced, therefore we do not have the vulva. Now the gene has another mutation Z which is again producing a mutant form of protein, that's why we do not have the vulva. Could you understand? It is affecting the same protein, or it is affecting the same gene. That's why they are in the same complementation group. So that's why we have there high chance that Y and Z can be on the same gene and they cannot complement each other. Yeah. Ajay, you have raised your hand. You wanted to talk to me? Okay, okay. So again, to emphasize, so the key point that I want you guys to learn and imprint it in your mind is that complementation group, then the phenotype, then the phenomenon is opposite. In a complementation group, nothing complements. Actually, it is very ironic. In a complementation group, nothing complements, while in another which are not in a complementation group, that may or may not complement, depending on the gene and pathway and everything else. But in a complementation group, they will never complement each other, right? And if you have any doubts on this, we can revisit this. But we have another question related to the same table. So YZ belong to the same complementation group, and they are on the same gene and cannot complement each other because the two mutations in the same gene will still produce defunct gene product. So here is another example apart from C. elegans to visually show you the two mutations. No complementation, two mutations in the same cistron. Cistrons mean the coding region. So uh, here we have a virus and a E. coli. We infect the virus on the E. coli, which will transfer its gene inside the E. coli. We have a bacteriophage which can infect a bacteria. So this is an assay in which bacteriophage carries our gene of interest, which can then which can infect the E. coli and integrate its genome inside the E. coli. Right? So what will happen? We have a during simultaneous infection, no complementation occurs because no functional A products are present. So the bacteriophage we have two genes A and B, and in the A we have mutations. So one kind of bacteriophage has this blue colored mutation and the other has yellow colored mutation. Now how would we know whether these two are from the same complement group? We do a simultaneous infection onto the E. coli. Now we see that mutation is on the same gene, then gene A will be still defective. Simultaneous infection, no complementation occurs because no functional A products are present. Now because no functional A products are present, there are no plaques. A is a gene, let's say antibiotic resistance gene, that let's say ampicillin resistance gene, which is going to confer ampicillin resistance to the E. coli. Now since there are two mutations in the A and they are in the same complementation group, they cannot complement each other and a defective antibiotic resistance gene is produced, thereby which we see no plaques. When we grow E. coli, this kind of E. coli in antibiotic plate, in ampicillin plate, it does not have resistance because both of the mutation form in the same complementation group and cannot complement each other. I hope with this example it is clear to the students the basic concepts of complementation. Just let me know in the chat box with a yes or no and we'll move on to the next question. Do not worry guys, we'll move very slowly and we'll clear all the concepts. I will try my best so that all the people attending this class would be would derive some benefits. There might be some things which I do not know, but if you have doubts in those areas, I'll get back to you. You can write to me and or you can pose a question. But for now, is it clear? Question 4, question 3 about the complementation group. You can just type yes or no and I'll move on to the next question. Okay, great, great, cool. So the fourth question, so the fourth question here has a twist. Another mutation arises, another mutation T. Now the mutation is T, was later identified, which also caused vulvaless phenotype. So T is another mutation causing vulvaless phenotype. A cross between TT and the other mutants listed above resulted in wild type progeny in all cases. So we have identified a new mutation T now, which is crossed to all those previous mutation V, X, W, Y, Z. And now we have wild type in all cases. So T was earlier a vulvaless phenotype, but now upon crossing with all these previous mutation, it is wild type. Now what can be deduced from this? What can be deduced from this about T, about this nature of mutation T? Options are T belongs to same complementation group as X, T belongs to same complementation group as Z, T is in its own complementation group, T is on same gene as W. You can see there are a variety of statements. 1 and 2 statements are about complementation group, the third is also about complementation group, and the fourth is about a gene. So you have to read these statements carefully and think about it. If you need the table, I can show you the table, but I think you will be able to solve it verbally, because now if you are clear about complementation concepts, let me know in the chat box what do you think is the correct answer. I'll wait for a minute and we'll move forward. Okay, Ajay is answering number 4. Other students, please try to answer. You can just guess on the based on your logic and understanding, it's fine. And if you have really understood it and are trying to solve the question, it's well and good. It's all for the better, right? Just write your options. We'll collect some more answers and I'll reveal the correct answer. Sakshi is saying 2. Simhasanam is answering 4. And Pushpanjay also answered 4. Yeah. Now the time is for revealing the correct answer. The correct answer is option 3. T in its T is in its own complementation group, and it is a little bit difficult to understand, might be for you guys. I'll explain it again. Let me explain with this table again. So here the earlier table we had, I have added one more column. We have the new mutation here, T by T, right? And we cross it with all these different mutations. In all the cases we get wild type. Even though T by T was a vulvaless phenotype, in all these cases we get wild type phenotype. This means that T does not belong to any of the complementation group in whichever these mutations are. So we had two complementation groups. Here you can locate them. V and X are one complementation group. Since crossing V and X, we still get the mutant phenotype vulvaless. That means they are not complementing each other. They are on the same gene. These mutations are on the same gene. They cannot complementation each other, and they form one complementation group because because of these mutations. These mutations create the mutant phenotype. That's how you select it. Similarly, W will form complementation group with Y and Z because they give vulvaless phenotype. Now if you see T forms no complementation group with anyone. That means T in itself is a complementation group. T by T is a third complementation group. So if you screen for more mutations, some other mutations might fall in complementation with T by T, but leading to the same phenotype, the vulvaless phenotype. This mutation is in a different gene, thereby not affecting the vulvaless phenotype. So that's how we come to the conclusion, because this is in a different gene. So mutation the VYV mutation will not affect the same gene. Now the both genes will have, when complemented, when coming together, both the genes will have their good copy, right? Both the genes will have their good copy, as we saw in the first explanation of the complementation. Since both the genes have good copy, they are not affecting the vulvaless phenotype. They are getting it wild type, and we are getting such wild types in all the cases. That's why T by T is not in complementation group with any of these mutations, and T by T also does not occur on any of these same genes. It will not occur with W, it will not occur with V, X, Y, Z, anything. It is on a whole different complementation group or in a gene. So this will be the correct answer. So now if I clear your doubts, most of the students get confused because why is it called a complementation group when nothing ever complements inside that group? It's very ironic, but let me give you a nice explanation for that. So here we see in a tabular form, it is well and good, it's looking nice, and we can identify with the wild type and the mutation phenotype. But when we actually do the crosses, when we actually do the experiments, when the scientists have identified, what do you think the scientist will pick up first? The scientist will eliminate and pick up the mutant phenotypes, right? Whichever genes is giving wild type phenotype, it's very vague to pick it up because any gene can give wild type phenotype. What is the alternative phenotype that we are getting? We get a vulvaless phenotype. So scientists are picking those groups, those worms, and then finding, oh, we crossed the V mutation with the X mutation and we get a vulvaless worm. But now wild type, you see, it can be vague. You cross V with W, it will still give wild type. You cross V with Y, it will give wild type. V with Z will also give wild type. So if you pick up a wild type worm from the plate, the scientists pick up a wild type worm, then it's very difficult to determine which combination of mutations cause this. But it is easier to determine which combination of mutation caused the vulvaless phenotype. That's why the scientists named this as a complementation group. But the characteristics are really opposite. Inside a complementation group, these mutations do not complement each other. That is why it resulted in a mutant phenotype. So that is a simple explanation of why this confusion is arising. You guys must have to remember that within a complementation group, the genes do not complement each other. I hope this is clear to the guys. So it's okay. We are all here to clear our concepts. It's good that we are dealing with such basic concepts in this problem solving session. If you are not able to understand, you can ask me. I'll clear it again for you guys. Is it clear to the students? Just let me know with a yes or no in the chat box, or you can speak up. It's okay. We have few students. Yes. Great, 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 great. Now let's move on to the next set of questions. So here we have a different table. Questions 2, 3, 4 we solved with one table. Here we have a different table. You have to read this question very carefully. Assume A, B, C, D are four alleles identified through a genetic screen of gene X in C. elegans. So there is a gene X and there are four alleles A, B, C, D. Gene X is known to produce protein P, which suppresses the production of protein Y, which induces vulva formation. So this statement is very important. You have to draw out the hierarchy. Gene X produces protein P, which suppresses production of protein Y, which induces vulva formation. So this is the statement, and this is the following observation that were made. Genotype AX, what is the effect on P, effect on protein Y, and what is the phenotype, resultant phenotype. Genotype AX, when allele A is with X, higher levels of P, effect on Y is absent, phenotype is vulvaless. Similarly, BX, effect on P, present as aggregations, effect on Y, present, phenotype multivulva. CX, effect on P is present, effect on Y is absent, and phenotype is wild type. DX, effect on P is present, effect on Y is higher levels, phenotype is multiviva, and CC, effect on P is absent, effect on Y is present, and the phenotype is multiviva. So you guys have to look at this table, understand the statements, and then I'll put up the questions. But to make it easy for you guys, since this is a problem solving sessions, I have drawn it out to for you guys to understand, because I want all of you to understand it very well. So this is the relation in the question here. We have a gene X which produces a protein P. Protein P inhibits the production of protein Y, while protein Y is responsible for vulva formation. Understood? This is the statement. Gene X known to produce protein P, which suppresses the production of protein Y, which induces vulva formation. So this is the relationship here. And here on the lower side is the table. So I want you guys to have a good look at this table for a minute or so. I'll stop talking and you guys can have a look at it. Then I'll pose the questions. So have you guys had a good look at this table? Let me know with a yes or no in the chat, and I'll start with the questions. Because the following two or three questions will be on this table. No screen. Oh, Ajay, are you not able to see the screen? Maybe I'll reshare it. Just a minute. Is it visible now, Ajay? And others also. Simasanam says yes, I think it's visible to him. Are you able to see? Okay, okay, great, great. Yeah, just have a quick look. I have drawn it out, the question for you guys. Right, gene X to protein P. Protein P is inhibiting or suppressing the production of protein Y, which is leading to vulva formation. Okay, so now we have the question. Question number 5, we have a match the following here. Based on the information in the table, what might be the allele types of ABCD? So we have four different allele types and four different alleles here. You have to match the allele type with the correct allele. We have four different allele types: antimorphic, hypermorphic, neomorphic, recessive, and on the left side, ABCD. I think it's easier to remember these four types, and then I can show you back the table for you guys to refer. Antimorphic, hypermorphic, neomorphic, and recessive. So let me do one thing. I'll type these four options: antimorphic, hypermorphic, neomorphic, and recessive. And we have ABCD in order. These are the order. So I'll switch on to the table now. You guys can refer the table and let me know your answer. This is the table. You have to match ABCD. Which of these alleles do you think is antimorphic, hypermorphic, neomorphic, and recessive? So there is a basic hint in the table that is provided to you. You can study the effect on different proteins here and the phenotype that it shows and correlate with the definitions of these types of alleles. Antimorphic, hypermorphic, neomorphic, and recessive. Take your time and think about the answer. Let me know in the chat once you are ready. You can post it in the chat and I'll reveal the correct answer. So for the options that we have here, I have changed it. Here are the options: 1C, 2A, 3D, 4B. Sorry. And 1B, 2C, 3D, 4A. So there are four options here. Ajay and Sakshi are both answering option 4. Okay, option 4 means D. Are you sure, guys? Because you might want to have a look. I know this question is a bit complex and we have a lot of tables here, but believe me, I was not able to accommodate the table and the question both in the same slide because the writings would have been too small, but you have to refer it again and again. So without any delay, we have two answers. So let me reveal the correct answer. The correct answer is option 2. So the option 2 has all the correct matchings. Gene A hypermorphic, allele A hypermorphic, allele B is neomorphic, allele C is recessive, and allele D is antimorphic. So these are the four different types of alleles we have. The answers: A is hypermorphic, B is neomorphic, C is recessive, and D is antimorphic. Now let me introduce you to all these four different kinds of alleles that we have encountered in the question. What is an antimorphic allele? An antimorphic allele, also known as dominant negative allele. So these terminologies are very important. You will encounter these in actual research papers and any research problem if you are going to use such alleles. Dominant negative allele, very common scientific lingo, is a specific type of allele that interferes with or counteracts the function of wild type normal allele even in the presence of one or more copies of the wild type allele. So dominant negative, just two words, antimorphic. Anti means against it. Dominant means it can override the effect of wild type, so it can dominate over the wild type gene, and it is a negative allele. It produces a negative effect. Counteracts the function of wild type even in the presence of one or more copies of the wild type. So this is a antimorphic allele. It will be dominant and it will have negative effect. Then a hypermorphic allele, yeah, A A you think is high antimorphic. Ajay, I will come to the explanation in the next slide. Just wait a minute. I'll come to the explanation. You can ask me then, or you can just use the chat box. Yeah. So as I was saying, a hypermorphic allele is a type of allele that leads to increased level of gene activity or an enhanced version of gene's normal function compared to the wild type. Hypermorphic, more product or more gene activity. So it's pretty easy to understand. Hypermorphic, more product activity. Antimorphic, dominant and negative, dominant and less activity. And then neomorphic allele is a type of allele that results from a mutation in a gene leading to new or altered function of the gene product compared to the wild type allele. So neomorphic allele, mutation which will create a new product altogether, right? Mutation in a gene leading to new or altered function of the gene product. And a recessive allele is a type of allele that expresses itself only when present in homozygous state. I'm not audible. Sakshi is saying that I'm not audible. Okay. I don't know. Maybe it is a problem for my internet connection. I have to check it again. Please repeat it again. Okay, so I'll repeat it again. The antimorphic allele. We have four different. Am I audible now, Sakshi, and the other students? Are you able to hear me? Yes. Okay. Ajay, are you able to hear me? And all the other students? Okay. Let me just go through it again. The antimorphic allele is known as dominant negative allele, specific type of allele that interferes with or counteracts the function of wild type normal allele, even in the presence of one or more copies of the wild type. So it will be dominant over the wild type and it will produce a negative effect. That's why it is antimorphic or dominant negative. A hypermorphic allele on the bottom left is a type of allele that leads to increased level of gene activity. So that allele will lead to increase in gene activity, or whether increase in protein production or whatever the gene activity is. Neomorphism, neomorphic allele means a new, neo means new, hyper means more, anti means negative, neo means new, a mutation that creates a new phenotype altogether or a new function of the gene product altogether. That is a neomorphic allele. And a recessive allele is a type of allele which will only express in homozygous condition, meaning an individual has two copies of the recessive allele and no dominant allele to mask its effect. In presence of dominant allele, a recessive allele will not express itself. Only in a homozygous condition, a recessive allele can express itself. So these are the four different definitions that we encountered in the match the following. Now let us understand the match the following here. I think doubts of Ajay will be clear here. So Ajay was saying that option gene A would be antimorphic. Let us see how, uh, but I think option A should be hypermorphic because gene A is increasing the effect on P. Gene A is increasing the effect on P, the higher levels effect on P, but effect on Y is absent. And we get a vulvaless phenotype. So because we have higher levels of P, if you remember the relationship, gene X leads to P, P leads to inhibition on Y, and Y leads to phenotype. Phenotype or the vulva formation. So higher levels of P, Y is inhibited more and there is no phenotype. No phenotype means no wild type phenotype. We have a mutant phenotype, the vulvaless. That's why A would be higher levels effect on P. A would be hypermorphic. Ajay, how were you saying that? Can I hear your part of the argument that A could be antimorphic? Yeah, even without action on Y, you were saying. Yeah, yeah. I think you are. Are you clear now, Ajay? That A would be. Yeah, yeah. Okay. And then B, if you see, effect on P is a different effect. Here we see this is present as aggregations. An effect on Y is present and we therefore get a multivulva phenotype. So you see there this is a different new effect that the allele B is bringing onto the gene X, right? So B is a neomorphic allele of gene X. And C, if you see for allele D, it has higher levels of effect on Y. So it is producing a negative effect and higher effect on Y because if you see here the presence of Y, so the presence of Y negates the effect of P. If you see these the relationship here, more the protein Y, the more the protein P, there will be less protein Y. So what it is doing is allele D is giving more protein Y, higher levels of protein Y, and effect on protein P is present. So that's why it's dominant negative. It is producing in higher amount and in a negative effect for gene X, not for the vulva formation. So you see, with respect to gene X, protein Y is negative impact, right? So with respect to gene X, these alleles are, we have to find out what in respect to gene X are the properties of this allele. So if it has a higher level effect on P, Therefore, A is hypermorphic, present as aggregations, which is a different new phenotype. So B is neomorphic. D have higher effect on Y, thereby opposing the gene X to protein 3 gradient. So we have D as antimorphic. And the last one, CC, if you see, if CC is present, then effect on P is absent, effect on Y is present, and we get multivalve phenotype. So CC here is a recessive allele which will only express in homozygous condition. So in presence of X, C was not expressing, effect on P was present, but when we have CC, the effect of P is absent. So this will be the correct answer. Yeah, Ajay, you wanted to say something? So indirectly X is having an effect on protein Y, right? Yeah. Yes. Yeah, yeah. Which allele are you talking about? B, present as aggregations and effect on Y is present. Hmm. Yeah. For gene A, you are saying? Yeah. Yes, 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 I understood. Higher levels of pre will repress the protein Y. Yeah. No, you see, if CC is present, that means gene X is having a repressive phenotype, means gene X is not expressing properly. That's why we see an indirect effect on Y. If you see the CC1, yeah, yeah, yeah. Indirectly, the protein P will influence protein Y. You, but high, yeah, yes, yes. They are producing P and Y effect through the gene X actually. These are alleles. These are alleles of the gene X. That's what if you see the question, assume ABCD are 4 alleles identified through a genetic screen of gene X. So all these alleles are affecting the gene X, and through that they are indirectly affecting gene Y. So we see the total effect. Okay. Huh. Actually, protein Y is necessary for vulva formation. So you see, wherever we have Y is present, we have multivulva, which induces vulva formation. I think the statement here means protein Y is what induces. Okay, okay, okay. I'm sorry if if that got confusing, but here as the statement says, protein Y is what which induces vulva formation. So the all these relations lead to protein Y influencing vulva formation. That's why we see wherever effect on Y is positive, we see multivulva. Effect on Y is higher levels, we see multivulva. And so on. Dominant negative, yes. Okay, okay, okay. Thanks for asking me and clearing your doubts. Now let's move on to the next question. Which of the following is a loss of function allele? So out of these, which do you think is a loss of function allele? If you we have to go back again to this table. Which of these is a loss of function allele? A, B, or C, or D? Come on, guys, you have to quickly answer because we have short time left after this, just 30 minutes. And Ajay has answered option A. Okay. And other students, which of these is a loss of function allele? Was the question. Can you identify which is the loss of function allele here? Okay, I think we should move on quickly. We should move on quickly to the answer. So loss of function allele, the correct answer would be option C. Gene C would be the loss of function allele. Loss of function alleles are often recessive. This means that the presence of functional allele can mask the effect of loss of function allele in heterozygous individuals. Right? So in presence of a dominant allele, the loss of function allele would not show its phenotype, but only in homozygous recessive type, the loss of function allele will show its phenotype. A loss of function allele is a type of allele that has undergone a mutation that results in reduction or complete loss of gene's normal function. So I think Ajay's answer is also correct, option A, but option A would be dominant negative form of the allele. So that is also a way of creating a loss of function allele. So in experiments when we create a dominant, we create a loss of function phenotype with dominant negative allele. But if you are going to talk about loss of function allele, it would be a recessive allele, which in case here is C. So we see CC has effect on P where it is absent, and thereby we have effect on Y and we have a phenotype, while CX here, in presence of a dominant X allele, the effect of C is not seen. But AX, which Ajay has answered, is a dominant negative or antimorphic, sorry, it's showing kind of antimorphic levels here, higher level and antimorphic. Is it correct? A is hypermorphic, sorry. D is antimorphic, yeah, D is having, A will also have, now that I see here Ajay's argument is looking kind of valid, because higher levels on P will effectively reduce the level of Y, right? That's why you have answered A, I think. And then we have a phenotype. But generally, for the answer to question 6, we have CC as the correct answer. C will be a loss of function allele, and it is mostly recessive alleles that will have loss of function phenotype when encountered in a homozygous state. Either complex is not formed or complex function is not performed. Now, since we have less time, I'll try to finish this session with other 3-4 questions. So question number 7 here, there are 3 statements, and you have to identify which of these are true or false. A suppressor screen is done on mutant backgrounds to identify new mutations that strengthen the mutant phenotype. This is a suppressor screen. Statement about suppressor screen. Synthetic mutations help identify genes that function in a single pathway. Synthetic mutation help identify genes that function in a single pathway, and hermaphrodites allow one to screen for effect on 2 chromosome sets in F1 progeny. Now, which of these statements do you think are true? You have to be very fast because we are almost reaching the end of this session and we have to finish 2 more questions. Which of these statements do you think are true? First statement is about the suppressor screen. Second statement about synthetic mutations. And hermaphrodites allow one to screen for effect on 2 chromosome sets in F1 progeny. Okay, Ajay have answered all are true. Just a minute. Just a minute here. Now let me reveal the correct answer to this question. The correct answer is statement 2 and 3 are true. Synthetic mutations help identify genes that function a single pathway, and hermaphrodites allow one to screen for effect on two chromosome sets in F1 progeny. Now let us learn a bit about all these three things. So first we have a suppressor screen. Suppressor screen is a genetic experiment conducted to identify second site mutations that can suppress the phenotypic effects of a primary mutation. It means whatever mutation, whatever the primary mutation is causing a phenotype, if we have a suppressor screen, we can identify other mutations which can suppress this mutant phenotype, right? It can suppress, it will help us to suppress that. So the statement one is a little bit change misleading here, because a suppressor screen is done on mutant backgrounds to identify new mutations that strengthen the mutant phenotype. So while it is the reverse, new mutations that can reduce the mutant phenotype or suppress the mutant phenotype, those kind of mutations are coming up in a suppressor screen. That is why statement 1 is not true. It's just the opposite. Any mutation which will strengthen the mutant phenotype, we do not get that in, we do not select for that in the suppressor screen. The primary mutation typically results in a specific phenotype which can be abnormal or detrimental to the organism. The goal of suppressor screen is to find additional mutations that suppress or reverse the effect of primary mutation. So it will reverse any mutation which can reverse the effect of primary mutation. And this is a simple method, step-by-step method of performing a suppressor screen. So first we have to identify the primary mutation. Start with an organism or cell line carrying a known primary mutation that results in a specific phenotype of interest. So uh, let's say mutation in a gene is causing loss of some nutrient or whatever phenotype, a big phenotype. Then we perform mutagenesis, introduce additional mutations into the genome of the organism which already has a primary mutation, and the goal is to create a population of individuals with primary mutation and various secondary mutations. We have various secondary mutation. Then screening for suppression, we observe the resulting population of individuals for any individuals that exhibit a reversal of the primary phenotype. Now we select such individuals in which the primary phenotype is reversed, and in such individuals we say that these individuals have acquired secondary mutation that can suppress the effect of primary mutation. Now we isolate these individuals and we sequence them, or we identify what are these specific secondary mutations which are responsible for this suppression. That's how we identify mutations which can suppress the primary mutation phenotype. So this is all about suppressor screen. And synthetic mutation, here is an example on my right about synthetic lethality. Synthetic mutations are powerful tool in genetic research that can help identify genes that participate in a single pathway or functional process. So this was a second statement. Synthetic mutation can indeed help us identify single pathway. So here if we have two genes, gene A and B, performing an important function which when ablated gives to lethal phenotype. So we have gene B mutated, one of these genes mutated, then we still have a viable phenotype. When we have gene A mutated, we have a viable phenotype, but when we have a double mutant, both gene A and gene B, this leads to lethality, synthetic lethality. This is an example of synthetic mutations. Synthetic mutations involve creating double mutants by combining two individual mutations, like here, gene A and gene B, that have different effects on the same pathway. So on survivability pathway, these have different effects, but when both of them are ablated, then it is lethal. The goal is to observe the combined effects of these mutations and deduce the relationship between the genes involved. So any two gene involved, the relationship can be deduced. If the phenotypic effect of double mutant is similar to more severe single mutant, the genes might act sequentially in the same pathway. So uh, this is an interpretation. If we have a single mutant phenotype, let's say gene B is giving some phenotype, and the double mutant is giving an enhanced gene B phenotype, then the genes might act sequentially in the same pathway. It means that gene A and gene B are sequential in the same pathway. If the phenotypic effect of double mutant is less severe than that of either single mutant, so compared to gene A and gene B single mutants, if the double mutant is having less severe phenotype, it means they are in opposing pathways. Like one pathway, gene A is involved in increasing the function, gene B is involved in decreasing the function. Both are in opposite pathways. So these kind of inferences can be made by making synthetic double mutations, by bringing them together. So option 2 is correct. They can be used to identify genes of the same pathway. And statement 3, where a hermaphrodite, so hermaphrodite basically means combines two sets of genomes from the same individual in the F1 progeny, because hermaphrodite is an organism with both male and female sex organs. So they will have both male and female gamete, and they will self-fertilize. A hermaphrodite can self-fertilize. On self-fertilization, so it can then bring together two sets of genes from the same individual in the F1 progeny. And the effect of two sets of genes can be studied in the F1. So the same individual will have two set of genes, the female gamete and the male gamete, and these both can self-fertilize and then can be studied in the F1 generation itself, the effect of two sets of genes. So statement 3 is also correct. I hope it is clear to the students. Statement 2 and 3 is correct. Statement 1 for suppressor screen does not enhance the mutation phenotype, rather than reduces it or suppresses it. Now we have another interesting question on the screen. Consider the following sequential metabolic pathway where M1, M2, M3 and M4 denote metabolites produced in the pathway, and A, B, C denote genes coding for enzymes that catalyze these reactions. So this is a typical metabolic pathway, and ABCs are enzymes, M1, M2, M3 are different metabolic states leading to the production of M4 as a final product. So guys, have a look at it. And then we have the question: when the gene B is mutated, which of the following is false? When gene B has a mutation, which of the following will be false? Now I want you guys to take a look at this and at the options and try to answer. When gene B is mutated, right? Option A, M2 is not produced. Option B, M3 is not produced. Option C, M4 is not produced. Option D, there is no effect. So M2 is not produced, M3 is not produced, M4 is not produced, and there is no effect. Which of these will be correct? Which of these is false, actually? Which of the following is false? Let me know in the chat box about what do you think is the correct answer. Simhasanam has answered 3. Ajay is saying M2 is not produced, which is I think option A. Okay. Others, do you want to attempt this? Okay, let me reveal the correct answer for this question. Pushpanja is answering B, M3 is not produced. So you have to read the question, which one of the following is false, right? The correct answer is M2 is not produced. I think Ajay has answered it correctly. This statement is false. And why so? So here, if you follow my representation here, we have a mutation in this B, enzyme B. So in this enzyme B, when it is mutated, M2 to M3 conversion, M2 to M3 conversion is halted. This will not happen. So there is no functional M3. Since there is no functional M3, there cannot be functional M4. So both M3 and M4 are not produced. But M1 to M2 conversion is functional because we do not have mutation in A. A is still functional. That's why M2 is produced. Right? If you now look at the options, which of the following is false statement? M2 is not produced is a false statement. M3 is not produced is a correct statement because B is the enzyme catalyzing M2 to M3 production. And M4 is not produced because we do not have the starting material, which is M3. So M4 is not produced, M3 is not produced, whereas M2 is produced. That will be the correct answer. I hope everybody is clear with this explanation. Or you are saying via production of, like, non-production of M4. Yeah, you're correct. Yeah, it's correct. You are correct. There would be no effect, but when in such a question, it's tricky when context is not given. Let's say in the question, if there was an additional statement, M4 is producing some function or doing something. It's a metabolite. I agree, but M4 is causing some phenotype or color or whatever such thing. Then we can easily say that, oh, there will be no effect since B is mutated, one of the enzymes is mutated, then we do not have any function. Yes. Yeah. Yes. You are correct. Option D will also be the correct answer. So the correct answer to this question is option A and option D. Let me mark it here. I absolutely agree with Ajay and his explanation. There is no effect will also be the answer to this question. Okay. Now let's quickly move on to the next question. What is not true about the epistatic nature of genes in this pathway? Now you have to figure out which genes are epistatic over which. We have four statement here. A is epistatic over B is epistatic over C is epistatic over B, and A is epistatic over C. These are four statements. What is not true about epistatic nature of genes in this pathway? In this pathway, readily from the previous solution. Now, what is not true about epistatic nature of genes in this pathway? A over B, A over C, B over C, and C over B. These are the four options. What is not true about epistatic nature of genes in this pathway? Yes, Ajay has mentioned option three. Yeah, Ajay, it would be better if you post it in the comments. And other students, what do you think is not true about these statements? 4 and 3. Simhasanam. Okay. Now let us see what is the correct answer. Option 3 is correct answer. Ajay has answered correctly. And Simhasanam answered 4 and 3. I think after this explanation you will be able to understand. Let us start with the definition of epistasis. Epistasis is a certain relationship between genes where an allele of one gene hides or masks the visible output or phenotype of another gene. So it doesn't have to necessarily be consecutive for the effect or for the epistasis. So A can be epistatic over C because A can regulate the output, visible output or phenotype of the C, enzyme C. So A is epistatic over C. The effect of gene mutation is dependent on the presence or absence of mutations in one or more other genes. So this is another representation of epistasis in which mutation in one or more genes can cause the effect in the downstream gene. So since A occurs in the upstream of B and C, any mutation in A will affect the visible product of C and B both. That's why we say A is epistatic over B and C. Whereas the reverse is not true. Any mutation in C will not affect enzyme B or the product, visible product, whatever enzyme B is doing. So C is not epistatic over B. So here is another easy representation. We have two genes located on pair of homologous chromosome. The gene B influences effect of gene A. Then the gene B is said to be epistatic to the gene A. If whatever gene B is doing is affecting the activity or whatever the conversion, the phenotype of gene A, then B is said to be epistatic over A. An unrelated gene influences the outcome of another gene. That's why we will have C over B is not epistatic while all other relations are epistatic. Even A over C will be epistatic because A can indirectly affect the visible output or phenotype of C. So the following relationship are epistatic. A is epistatic over B and C, and B is epistatic over C. This will be the correct answer. A over B, A over C also, and B over C. But the reverse is not true. C cannot be epistatic over B. I hope it's clear. Okay, okay, yeah. Yeah, I understand. So M1 is the starting material. Simhasanam, did you clear your understanding here about epistatic genes? Okay, okay, great. Now for the last question, question number 10. Let us assume that there exists another arm to this pathway, whatever we were having, that allows M2 to be converted to M4 via M5. So we have another branch here. Through the M5, we can go to M4 now. Now, which of the following is correct? Statement 1, just single mutations can no longer reveal sufficient information about all epistatic relationships. Statement 1. And statement 2, mutation of B will show a phenotype. These are the two statements, and you have lot of options. Statement 1 and 2 are true, and statement 2 is the correct explanation of statement 1. Statement 1 and 2 are true, but statement 2 is not the correct explanation of statement 1. And then 1 is true and 2 is false. 1 is false and 2 is true. Both 1 and 2 are false. Now let me know quickly, what do you think is the correct answer? Okay, Ajay have answered statement one, the option one. Statement one and two are true, and statement two is the correct explanation of statement. Okay, let me just reveal the correct answer. The correct answer is statement one is true and statement two is false. So what we have here, just single mutations can no longer reveal sufficient information about all epistatic relationships. This statement is true. Mutation of B will show a phenotype is not true. So for both these statements, we refer to this figure. Yeah. Yes. Yes. Yeah. Yeah. I understand. But in such questions, you have to. Yeah. 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 I understand. I understand. But for questions like these and the other questions where you had confusions, you have to, there are certain assumptions that these questions make. I know this is very difficult, but a general notion that follows, there are no proper instructions, but a general notion follows that when we say a phenotype here, it means a mutant phenotype. Actually, the one that you answered based on M4 creating a phenotype is actually very correct. But here what the question means, which is not said in the question, is a mutant phenotype. So mutation of B, will it show a mutant phenotype? No, because mutation in B will, no, mutation of B will, the, Yeah, yeah, yeah. That means if one of the pathway here is impaired, let's say we do not have B, we can still reach M4. So I apologize, but the questions you may face questions like this in your exams, where you have to assume a lot of things. Let's say the one that you said, that right to left, we do not know which is the starting point and which is the end point, and really is a metabolite giving a phenotype, or is it just mentioned the way it is, and what do they mean by phenotype? Is it a mutant phenotype? Is it the final phenotype? Or is it the same mutation in the gene causing a phenotype? Right? So questions can be confusing. So you have to, I seriously have no clue how the questions will come, but you have to use your presence of mind and find the best answer. But these are previous year sample questions, so I am solving it here. So with this, we have come to the end of the presentation. So this is what I meant. Mutation of B will not show a phenotype it, with respect to M4. That is the answer. You are absolutely correct, as well as other students also. There exists an alternative pathway via M5 to reach the final outcome by N4. B is only epistatic over C. So mutation in B will not affect E, which can still lead to M4. So in your ways, you are correct, and but the answer that I have been given with is statement two is false. Interpretation can be a little different, and the questions should be more elaborate. I understand. So with this, we have come to the end of this session. I hope you guys could take back something, and were able to understand the actual problem solving sessions would really look like this, because there is a two-way interaction and we solve a lot of questions. There can be different answers, there can be different arguments. So since I'm a TA, you can interact with me freely. You may not be comfortable in uh, solving these things, but now you'll be more accessible to these concepts and clear your concepts and keep excelling in this course. So for what I'll do here now, for the students that are here, I'll paste again the YouTube channel link. You might have already have the access to the slide presentation and YouTube channel for these sessions. This is a recorded session, so I'll just quickly upload the YouTube channel. For your information, guys, I also had a brainstorming session with you guys, and lots of discussion, and with your clearing your doubts and clearing my own doubts. So we are all growing together, and it's really good to know that. And I would encourage all of you guys and ask your friends and join the TA sessions. If you missed mine, then you can join Partha days, and you should both simultaneously join both. So it will be more experience for you guys, right? So let me just put in the playlist. So I've created a playlist which contains the whole recorded series for developmental biology. No, Ajay, not any group I have been mentioned of. So if you see this NPTEL courses and yeah, yeah, I understand. Yeah, I understand, but I think NPTEL is providing you with the links and all. So yeah, okay, okay. So are you a student at IITK or IIT Madras? Okay. Yeah, I think the presentation links and YouTube links is all that I can do for now. Let's meet over the weekends. And yeah, yeah, I'm just putting this YouTube link here so that students who are attending this can see it. You might be, you must be getting NPTEL emails where you have the YouTube link and the presentation folders also. So I'm just separately sending it so that people can easily access it. So you already have that kind of information. Every yeah, yeah, I understand. Mm. Yeah, yeah, yeah. Okay. So I'll end this meeting, guys, and thank you for joining. See you in the next week for week 5. Bye bye.